Now we are going to look at the slides related with the male reproductive system and continue to our lecture. Here we can see the anatomy of male reproductive system. We talked about the testis and the tunica albumina present in this part. As you know from your anatomy lectures, testis present in the sacrotum, and sacrotum have a couple of layers from outer to the inner side. You know these layers from the anatomy. And we will talk about the epididymis and ductus deferens and also the different secretory glands related with the reproductive system and also the penis. You can read this part for yourself. We talked about the lobules and lobes of testes. Here you can see a section from testis. This part is the mediastinum testis. Here you can see the septas and you can see the lobules in the inner side. This structure is seminiferous tubule, and between the seminiferous tubules we have interstitial area. Here you can see a section from seminiferous tubule again. This is the seminiferous tubule. Here you can see another section, and this is another section. This part is interstitial area. Here you can see the Leydig cells in the outer side of the seminiferous tubule, and in this side you can see the myoid cells present in the tunica propria, the spermatogonias in this part, and primary spermatocyte present here. Here you can see an example for Sartoric cell. You can see the late spermatids or spermatozoas in this part, and also you can see the early spermatids. Let me show you. Not early spermatids present here. Here you can see another section from mediastinum area. This is the mediastinum part. You can see connective tissues in this side, and here you can see the inner side of the lobules. This structure related to the seminiferous tubule. Here is mediastinum, and in the mediastinum we have the rete testis. You know the tunica vaginalis from anatomy. It has the parietal and visceral layer and a part of periton which continue into the scrotum. And also we have the punctiliform venous plexus. What is the punctiliform venous plexus? From your anatomy lectures, these venous plexus have a role in decreasing the temperature of testis from 37 to 34, because the testis need 34 degree of Celsius to work in normal condition. And we have interstitial tissue. In interstitial tissue, our most important cells is Leydig cells or interstitial cells, which have a role in the production of testosterone or dihydrotestosterone. And as you know, luteinizing hormone or LH have a role in the induction of Leydig cells and start the production of testosterone. The other name of the LH or luteinizing hormone in males is interstitial cell stimulating hormone. And seminiferous tubules, as you know, continue with straight tubules, and then rete testis. The rete testis connected with the efferent ductus and then connected with epididymis. We talked about this part. In the wall of seminiferous tubules, as you know, we have the germinal epithelium or spermatogenic epithelium. In outer side we have myoid cells. In the wall we have the Sartori cells and spermatogenic lineage. Here we can see different shapes of the seminiferous tubules. You can see the seminiferous tubules in this part and different cells in the wall structure. What do you think about these cells? This cells is our Leydig cells present in the interstitial area, and because of the high amount of lipid droplets in their cytoplasma, you can see foamy structure like which cells. In which cells we have these foam structures. In theca internal cells, in female reproductive system. In the cytoplasma of the cells of the cortex of adrenal gland, we have same structure cells. We have the spermatogenesis, spermiogenesis. We talked about these structures, spermiogenesis. Here you can see the same stages. And in this part, what is the name of this stage? Spermiogenesis, formation and transformation from round shape to line shape. Here you can see in the end of the spermiogenesis, we have a sperm or spermatozoa with head, midpiece, and tail part. And in the beginning, as you can see, the organelles migrate to the opposite side of the developing of acrosomal cap. And as you can see, this in this part you can see the growing of tail. And here again you can see the residual bodies detached from the cytoplasm of sperm, and in the end part you can see normal structure of sperm. Again here you can see this seminiferous tubule wall and different stages set. This cells is our spermatozoas or late spermatids. Here you can see the early spermatids, and in this part, for example, we have the primary spermatocyte or another primary spermatocyte present here. What is the name of this cell? Spermatogonia, because it's near to the valve. Type A spermatogonia, type B spermatogonias. We talked about this part and different stages. We talked about spermiogenesis, and during the spermiogenesis we have our acrosome form, flagellum form, and we have different phases, the Golgi phase, cap phase, and acrosomal phase, and maturation phase. You should know the events in different phases separately. Please read this part and memorize these stages. Sertoli cells, again we talked about Sertoli cells, have a role in formation of the blood-testis barrier, have basal compartment and abdominal compartment. What is the roles of the Sertoli cells? Supports protection and nutrition of the developing spermatogenic cells. They have exocrine and endocrine secretion like androgen binding protein, inhibin and Mullerian inhibiting substance production. And also have a role in phagocytosis. After testes, we have the ducts. The ducts divided in two types, the intratesticular ducts and extratesticular ducts. The intratesticular ducts composed of the straight tubules, retetestis, and ductuli efferentes. And here you can see section from the mediastinum testis. In this part we have the seminiferous tubule, continue with the tubuli recti or straight tubule. In the wall of straight tubules, we have only Sertoli cells. The straight tubules continue with the retetestis. In the wall of retetestis, we have simple cuboidal epithelium, and in the outer side, we have irregular dense connective tissue. Here again, you can see the retetestis, and in the outer side, we have the irregular dense connective tissue. Retetestis, again, here you can see. How we can understand this is a section from retetestis? Because this epithelium, simple cuboidal epithelium. This retetestis continue with the efferentes ductus or ductuli efferentes. Ductuli efferentes have a special shape. You can see these dentation areas in the ductuli efferentes. Because in the wall of dactyly efferentes, we have the shorter cells and longer cells, simple cuboidal and columnar cells present in the wall of the ductuli efferentes. Because of these differences in the age of the cells, you can see this special shape in the wall of ductuli efferentes. But in some other structures like ductus deferens and glands of the prostate, you can see same shapes. How we can understand? This is a section from ductuli efferentes. You should look at the basement membrane. In the wall of the ductuli efferentes, we don't have any mucosal fold, and we have only the cells of epithelium in this indentation area, and connective tissue present in the outer side, not inner side of the folds. Okay? This is the main differences. This slide is very important. You should memorize this slide. And here you can see the different types of the epithelium. In seminiferous tubule, for example, we have special epithelium. In straight tubules, we have Sertoli cells in proximal part and simple cuboidal cells in distal portion of the straight tubules. In retitestis, we have simple cuboidal cells with microvilli and single cilia. In ductuli efferentes, we have simple cuboidal and simple columnar ciliated cells. It's very important. In the wall of epididymis, also in the wall of ductuli efferentes, we have thin layer of smooth muscle cells. Very important. And loose connective tissue. In the wall of epididymis, we have the pseudocertified columnar epithelium with small basal cells and principal cells. The basal cells and principal cells, two types of the cells present in the wall of the epididymis. And as you know, epididymis is composed of the head, body, and tail. In the proximal side of epididymis, in the wall of epididymis, in outer side of the epithelium, we have the circularly smooth muscle layer. But this circularly smooth muscle layer in the initial part divided into the inner circular, outer longitudinal layer. In the we have the smooth muscle layers in the initial part, but in the end part of the epididymis, we have thick layer of smooth muscle layer, inner circular, outer longitudinally arranged smooth muscle layers. In the end of the tail part, but in the head, body, and proximal part of tail, we have only thin layer of smooth muscle cells. Don't confuse it. When we look at the ductus deferens, we can see the pseudocertified columnar epithelium with fewer sterocilia than epididymis. And also, in the wall of ductus deferens, we have thick layer of smooth muscle layers. In the inner side, we have the longitudinal, in middle side we have circular, and in outer side again we have longitudinally arranged smooth muscle cells. And in lamina propria of ductus deferens, we have high amount of elastic fibers. Because of this high amount of elastic fibers, we told this lamina propria is a type of fibroelastic lamina propria. Okay? It's very important. And also we have the ejaculatory duct, end piece of the ductus deferens in the inner side of the prostate. These ejaculatory ducts formed with the pseudocertified and simple columnar epithelium and have fibroelastic tissue in the connective tissue or lamina propria and also have smooth muscle cells. These smooth muscle cells related with the prostate stroma and not completely surround the ejaculatory duct. Can you understand this part? It's very important for your exam. It's very, very important. Please memorize this part as you need. Now we are going to talk about the extra testicular ducts or extraurogenital ducts. In this part we have the epididymis, ductus deferens, and ureter. As you know, we have the head, body, and head in epididymis, and in the epithelium we have the principal cells with stereocilia. This is lung cells, and also we have the basal cells near to the basement compartment. Spermatozoas in the inner side of the epididymis can develop and complete their forward motility. It's very important. And also, final modification occur in acrosomal part, and most important thing which occur in the epididymis is the capacitation. What is the the decapacitation, not capacitation. Capacitation occur in which area? Occur in the vagina, capacitation. Decapacitation occur in the epididymis. In decapacitation, the sperms lose their capacitation, lose their ability to fertilization of oocyte. But in the inner side of the uterus, again they can catch the capacitation ability. Okay, here you can see a section from the wall of epididymis. This red structure is our thin smooth muscle cells layer. Here you can see pseudostratified columnar epithelium of the wall of epididymis with stereocilia in this part, and in the luminal side you can see the spermatozoas. Again, here you can see spermatozoas in the luminal side, and this structure is spermatozoas. How you can differentiate the epididymis from seminiferous tubules? How you can understand this is a section from epididymis, not seminiferous tubule? Look at here, we have only the nuclei of cells in the basement part. Here we don't have the nuclei. Sperms completely separate from the epithelial cells. And also here we have the stereocilia in the epithelial cells. And here we can see the principal cells and basal cells. In ductus deferens, we have a structure like this. This part is our mucosa layer. In the mucosa, we have the lamina propria in this part and epithelium in this part. Here you can see. Again, here you can see this dentation, like ductuli efferentes. But here, as you can see, the connective tissue enter into the mucosal fold. But in the ductuli efferentes, we don't have connective tissue in the indentation areas, and we have the basement membrane which separates the epithelium from the connective tissue and not folded into the inner side. But here we have the folding. This is the main differences between the shape of the ductuli efferentes and ductus deferens. Lamina propria have a fibroelastic structure, and as you can see in the wall of the ductus deferens, we have the inner and outer longitudinally arranged and middle circularly arranged smooth muscle cells. And we have the ejaculatory duct which connected with the vesicular seminalis duct and continue with the prostatic urethra. We will talk about the prostatic urethra and accessory glands. Now we are going to talk about accessory glands. Accessory glands divided into the seminalis vesicle, prostate gland, and bulbourethral gland. Here you can see these glands and structures. Before the ductus deferens entered into the prostate, you can see the vesiculus seminal vesicle gland, and here we have the prostate gland, and in this part, in the area of membranous urethra, we have the bulbourethral glands. Bulbourethral glands have a shape similar with mucous secreting gland. Here you can see this shape, mucous secreting gland. But the vesicula seminalis and prostate have a little different structure. Here you can see a section from vesicula seminalis. In the wall of vesicula seminalis in high magnification, you can see the mucosal folds, and these mucosal folds form anastomoses with neighbor mucosal cells. And in the wall of the mucosal folds, we can see the simple columnar epithelium. In some areas we have the certified epithelium. And in lamina propria, we have connective tissue with high amount of smooth muscle cells. Look at here, you can see smooth muscle cells in the mucosal fold. In after side, again, we have smooth muscle cell layers, inner circular, outer longitudinal, or outer inner circular, yes, and outer longitudinally arranged smooth muscle cells. What is the function of the vesicula seminalis? The production of fructose, prostaglandins, and fibrinogen. What is the prostate gland? Prostate gland have the peripheral zone, central zone, and transitional zone. In peripheral zone, we have 70% of main glands of the prostate gland. And also we have the central zone, we have 25% of prostate in this part. And the glands in this part have a structure like submucosal glands. And also we have transitional zone, similar to the central zone, with high amount of mucosal gland. Our main structure present in peripheral zone. Look at here, we have the peripheral zone, the central zone, and transitional zone. In transitional zone, you can see glands generally related with the prostatic urethra. But in transitional zone, we have the main glands of prostate. In prostate, we have corpora amylacea, or prostatic sands in the lumen of the prostate gland. Look at here. This is a section from prostate. This is our prostatic sands present in the lumen of the glands. And in the wall of glands, again, you can see the columnar epithelium in some areas, and in some areas you can see the pseudo-stratified epithelium. And because of that, again, maybe you can see the similar shape with ductus deferens and ductus efferentes. And also, you can confuse in this part with the vesicula seminalis. But the main thing in separation is corpora amylaceae, and the epithelium of prostate composed of simple columnar and pseudo-stratified epithelium. In bulbourethral glands, as you know, we have only mucous secreting cells. And the last structure which we are going to talk about is penis. Here you can see a schematic drawing from penis in transverse section. As you can see, in outer side we have the skin. Under the skin we have the connective tissue, dermis of skin and hypodermis. And we have the dorsal veins and dorsal arteries. in this part. In the middle piece, we have three different structures. In upper side, we have corpus cavernosum, and in lower side, we have corpus spongiosum. In the middle of corpus spongiosum, we have the penile or spongiuretra, and corpus cavernosum and corpus spongiosum covered with tunica albuginum, irregular dense connective tissue. In the inner side of the corpus cavernosum and spongiosum, generally corpus cavernosum, we have these structures, trabecles which composed with connective tissue and smooth muscle cells in these areas, cover the cavernous sinuses. In the wall of cavernous sinuses, we have endothelial cells, and just beneath these endothelial cells, we have high amount of smooth muscle cells with central nervous system impulse. These muscles in the wall of endothelial cells relax. With this relaxation, the blood can deposit in the inner side of the sinuses, and with deposition, erection can occur. After inhibit the stimulation from central nervous system, the muscle cells again contract in this part, and with this contraction, blood return to the normal blood circulation and erection is ended. Here you can see corpus spongiosum. In the middle side of corpus spongiosum, we have the penile uretra. We talked about the penile uretra in our lecture about the urinary system. Do you have questions about the male reproductive system? Thank you for your attention.